QIMR Berghofer acknowledges the traditional and cultural custodians of the lands, waters and seas across Queensland, and we pay our respects to elders past and present. We support the cultural knowledge, determination and commitment of First Nations communities in caring for health and well-being for millennia.
It is a sickening blow when a child is diagnosed with brain cancer, and most of us know recurrence is the major concern, but less well known are the possible and permanent effects from radiation therapy. And Professor Bryan and his team and a collaboration from all around the world have a likely breakthrough on both of these fronts.
This is BodyLab, a QIMR Berghofer podcast.
Thanks for coming in, Bryan.
Thanks, Claire. How are you?
I'm very well, thank you. Now, congratulations. This is a great result from a huge collaboration.
Thank you so much. Yeah, it's very rewarding for us personally, obviously, as scientists, to bring these long studies together. And particularly in this case, it was sort of a bit of an interesting story, really, about how the project was initiated. But we were working with a relatively small drug company in Texas called Katana Pharmaceuticals. And they had developed this small molecule targeting OLIG2, and I'll talk about that in a little bit more detail later. Really, these drug companies, if they don't have any capital, what they do is they essentially farm the drug out to academic laboratories. And it had turned out, not in secret, but just it's confidential when they do this, that they pass it out to different labs. The CEO of this small drug company had actually set up collaborations with key labs all around the world, including us here in Australia, a very well-known group in Toronto at Sick Kids, a guy called Peter Dirks, Tim Gershon at Emory, and another lady in Washington. And so there was sort of 4 groups around the world working on this drug in parallel, but we didn't know it. And so we kind of thought we all had unique access to this drug and were working away. You know, it was really only sort of 5 or 6 years into the project through communication at conferences and things that we started to work out that we'd all been working on the same small molecule and we all had very similar overlapping data. At that point, scientists being scientists, we were chatty and sort of started to talk and show each other the data. Professor Tim Gershon at Emory and myself decided to team up and combine our data together just to, I guess, make it a more comprehensive story. At the same time, we're discussing with Peter Dirks, who had a parallel study in Canada. So, yeah, that's how the story sort of came to be. And so, yeah, the 2 papers ultimately were published side by side in Nature Communications, which was really, serendipity in a way that we got there.
And Peter Dirks really focuses on the biology of it. So they're complementary studies. You weren't doing the same thing.
No, I guess Peter Dirks is very well known as a stem cell guy, cancer stem cell guy. And so, yeah, he was one of the sort of doyens, founders of the cancer stem cell field in brain cancer. And so, yeah, he was really focused in that sense, trying to understand this protein OLIG2, which it's well known. It's a transcription factor which we know is involved in oligodendrocyte maturation or lineage potential. But we also know that this particular protein is re-expressed in, they believe, 100% really of adult brain cancers. We seem to be able to detect OLIG2 protein. And it's also highly expressed in medulloblastoma, which was the focus of our study, and also in DIPG, which is a very aggressive brainstem glioma in young children. And so it's kind of an obvious target for people in the field to look at these key lineage proteins, and OLIG2 is sort of one of these central proteins. And then I think having a small molecule that can target a transcription factor is actually quite a big deal.
That's a big deal because of the blood-brain barrier, right? So a lot of drugs that would be useful don't make it across that blood-brain barrier, but the size of the molecule of CT179...
Yeah, it crosses the blood-brain barrier. And so I guess the way I try to simply describe the blood-brain barrier is obviously you have a lot of blood vessels in your brain, which is essentially a tube, but then you have almost another tube of cells surrounding those blood vessels called pericytes. All those cells, if you can imagine linking together, they have tight junctions between those pericytes, and that biology is what regulates this blood-brain barrier. And it's essentially a protective mechanism to stop small molecules that could be damaging getting into your normal healthy brain. But obviously in the context of brain cancer, it also stops a lot of the current treatments we have. It's a sad story in a way, in that in the case of brain cancer, we've conducted 1,200 or more now clinical trials around the world, and they've essentially all failed. But unfortunately, many or most of those drugs weren't designed to cross the blood-brain barrier. They were repurposed drugs taken from other cancers. And so we've done all these clinical trials where many instances we're trying the drug in the veins of these patients or in tablets, but they were doomed to fail right from the start because we just are not getting enough drug into these patients' tumours in the brain to actually work. And it's taken a really long time for the field to, I guess, come to that realisation. And that's a complex question because it involves industry and, a whole lot of reasons for why that might be. But essentially now, finally, there's that understanding that we need to kind of slow down and redesign the drugs better to cross the blood-brain barrier. And that's exactly what this CT179 OLIG2 inhibitor does. We know it's the right size, it's the right polarity, and we know it's able to cross this blood-brain barrier and get into these tumours, and actually persists for quite a long time in the brain, more so than in the plasma or in the blood. And so that's really interesting to us.
So the point of CT179 is to inhibit OLIG2, and OLIG2 is the one causing the problems.
So OLIG2 is a stem cell marker. The best way to think about solid tumours is that they're obviously what we call heterogeneous. There's a lot of tumour heterogeneity. But put another way, there's lots of different types of tumour cells. Some of them are very early stem cells, and sort of almost like a throwback to development, in that you've got these early stem cells and then they actually go through differentiation inside the tumour and create different types of tumour cells. And so one of the unique abilities of cancer stem cells is they can go to sleep and they can hide out. The term for this is quiescence. And so there's been a really strong focus recently on quiescence, because we know what happens is when you use chemotherapy and radiotherapy, what they do is they essentially hit the replicating cells. So the cells that are quickly dividing, for the most part, that's how chemotherapy and radiotherapy work. They integrate or damage DNA, and then the cell tries to replicate again and dies.
And the stem cell then just hides and then decides at what point to come back, and that's the recurrence. And that's really where the danger is with brain cancer, particularly in paediatric brain cancer, which is your focus.
Yeah, that's right, Claire. I guess we know now in these very aggressive cancers, including brain cancer, that many instances, the case of brain cancer is very sad, but more than 90% often of these patients, the tumours are coming back. And so that's a big challenge. And we know that it's even tougher in that the current standards of care, being chemotherapy and radiation, kind of change the tumour, right? They kind of make it even more aggressive. And you've created something that's now resistant to the current standards of care. It's learnt to adapt to that. And it's also enriched for these stem cell populations and takes off. And so, that's why we're excited, I guess, about CT179, because it's really a way to target the tumour at its roots. We're trying to get at the fundamental cells that we believe are responsible for the development or initiation of the disease, and also the recurrence of the disease.
So what you're hoping is that you can use it in combination with radiotherapy and chemotherapy and then delay recurrence, and eventually down the track, is there a brighter option for that?
That's a great question. One of the things with young children is we have to give them relatively high doses, particularly of radiotherapy, to get a response. And we know this is very damaging. And it's very heartbreaking. If you look longitudinally, when they say a patient is cured, what they really mean is that they've had 5 years of disease-free status. And so you look longitudinally at these young children and, they're impaired, they don't leave home, they don't have relationships, they don't go to university, they often have cardiac problems. So there's a whole lot of negative impacts on their health from the standards of care. One of the big strategies that's really been looked at is to try and reduce, or the word we use is de-escalate, radiotherapy treatment in young children. And if we could keep the response the same, but lower the radiotherapy and combine it with another drug, we could keep the same survival but improve quality of life. And so that's what we're focused on. And so I think coming back to your initial question of where CT179 might work, I suspect that initially it will be used as a combination therapy, and we've got good data to support that, to show that CT179 we can combine with radiotherapy effectively, and it seems to increase the response. But we're also excited really about that opportunity of, as we discussed earlier, targeting the tumour at its roots and getting at those stem cells. And so that's one of the things that Peter Dirks is very strong on, the Canadian cancer stem cell researcher, in that if we can hit those cancer stem cells early enough in the disease, then we might really delay tumour formation very significantly. And so looking forward, if this drug is well tolerated, then there would definitely be opportunities to try that in the first line setting.
So would it help young kids with brain cancer to be diagnosed early, or is this more about recurrence?
That's a really interesting question, and one that the field and we think about quite a bit. Brain cancer is an interesting problem in that often it hides out and doesn't show symptoms until it's quite advanced. And so you often hear, that's a very typical patient journey, is they're completely normal, and then one day they wake up and they feel dizzy when they get out of bed in the morning, or they fall down in the shower and they go and have an MRI and they've got a very advanced tumour. That's the way it presents both for adult and children. And so right from the start, we're in a complex place with brain cancer patients. They've already got advanced disease. And then sort of the standards of care happen quite quickly, which involves sort of surgical resection. We try and debulk the tumour via surgery. That can happen really quickly for a brain cancer patient. Literally, like, you can wake up one morning, you'll feel unwell, you might have some dizziness, a spell, you fall down in the shower, as I suggested earlier, as an example. You go to the doctor, you get an MRI, and then literally within days you're on the operating table having your tumour resected, because it moves that fast. That the standards of care, we're trying to debulk as quickly as we can. And then there'll be sort of radiotherapy and chemotherapy regimes after that. So it's a really great question that if we could detect the brain tumours earlier, then obviously we'd have a much better chance, a much better shot at making patients live longer, but also the quality of life piece as well. And so one of the things that we and others have been doing is looking at biomarkers. And so it's a whole field of research where people are looking very much in peripheral blood to look at what you can find in peripheral blood of patients. And there's kind of three areas that people are looking at in the context of cancer. They're looking at the actual tumour cells themselves can break off and circulate around your body, so circulating tumour cells. The other one is exosomes, which are little fragments that come from tumour cells. And the other one is ctDNA, so like sort of tumour DNA can also be detected in blood. And so these are the three viable ways that scientists are trying to detect tumour cells early to give us that step up in terms of treatment. And so we have worked on those projects, collaborated on with a number of people. We've collaborated on a circulating tumour cell project, which interestingly, you can actually detect tumour cells and isolate them from brain cancer patients. Even though there is a blood-brain barrier, there still seems to be a mechanism by which brain cancer cells float around your body.
Yeah.
And we can pull them out of your blood, and we've actually grown them in the dish and proved that they are tumour cells. This is still early days, but looking forward into the future, the whole biomarker field hopes that we can do regular blood draws to detect. You go and have your bloods done once a year or twice a year. We would do some testing for potential cancer in your body somewhere, and if it lights up, then you have to go and have more tests. And
that is so exciting. But do you generally do blood tests on young children?
No, you don't. And so that is a very good point. I mean, obviously in the case of adults, that would potentially be viable. With children, yes, you're right. We don't do those sorts of tests. Children are by and large very well, and they don't get sick very often. And brain cancer in children is still rare. The biggest killer of children is accidents, unfortunately. And then the second biggest killer is often brain cancer. The reason that is true is because we haven't been able to cure the disease yet.
It really is exciting, and there's so many implications for where this research can go. Medulloblastoma is where you started, but there's opportunities down the road for glioblastoma. Tell us these results. I think you've played it down just how exciting they were in terms of what CT179 can do.
I guess, honestly, if you ask me to place where CT179 was, I think it's at the forefront, and that's the exciting bit for us. In a way, this work leads the charge of the new classes of drugs that I know are coming. And so we're kind of one of the first cabs off the rank, one of the first groups to really start pulling these brain-penetrating small molecules through the pipeline. And so that's the really exciting part for me, is that obviously we want to try and get these therapies to both children and adults as quickly as we can, get better therapies that cross the blood-brain barrier. So that's the really exciting bit. Finally, we are getting to the point, because I've been around in brain cancer for a long time. I've been in brain cancer space for almost 25 years now. I can't believe that, but it's true.
And just this last study was 5 or more years longer, wasn't it?
Correct. Yeah, I think all told there's probably 7 to 8 years worth of work that we've been working on this, but obviously much more for the drug company. They've probably at least another 10 years before that. So that's why these things take time to mature. But yeah, it's really exciting now to see. And I think the other thing that's really great about this study, as I suggested earlier at the top of our discussion, was really the collaborative nature. But what the collaboration does more than anything is, me as a scientist, I'm always worried that are our results right? Okay, like, are we seeing what we're seeing true? And you can get tricked up many times with biology because it's so complicated. But if the guys in Sweden are looking at the drug and they show it inhibits OLIG2 really nicely, and then Peter Dirks is over in Canada and he's looking at the biology and it shows it inhibits OLIG2, and then Tim Gershon is over in Atlanta at Emory using different models to me, but all using the same drug, and then I'm over here in Brisbane, we're all generating the same data, that makes me sleep well at night. It makes me think, well, hell, we might just be right. That's the exciting bit. Like, for me as a scientist, is I want other people to validate what I am generating in the lab, what we are doing. And that's the beauty of this big collaborative study. It's sort of, you imagine there's been there's actually 4 groups have independently validated this data over a long period of time. So it gives you that extra, for me personally, it gives me that extra sense of confidence that we're on the right track.
And I bet you can't wait for somebody to tell a little child with brain cancer or their parents, like, it used to be really damaging to the brain, the treatment, but now we have a better treatment and the chances of side effects from the therapies is going to be much lower.
Well, that would be obviously wonderful, and that's what we're working towards. That's the end goal, is, we want to, be able to really improve the outcomes and quality of life for these kids with brain cancer. It's truly terrible. It's truly terrible to witness, and you can't imagine what the families and the parents and obviously the children are going through. And so, yeah, you're right. I think we want to deliver on the promise. I guess that's at the forefront of our mind, is that obviously the community and the industry and the funding bodies and everyone, they want us to be successful. They want us to deliver these drugs. Obviously, everyone's been working really, really hard, super dedicated. Scientists, clinicians, neurosurgeons, all the funding bodies, the foundations, everyone. If you look at brain cancer, I know a lot of these people around the world. It's a huge effort has gone in. We're building momentum, and you need that breakthrough, I think, to keep people going and excite the field and keep the momentum moving.
The next step is really expensive and you need help. That is clinical trials, trying it in humans.
Well, that's the hard step next.
Yeah, you're in the valley of death, the scientists call it, don't they?
It's one of the valleys of death, yes. It's, we now want to get from the lab where we're excited and we think we've got something good. We've got full support from the drug company. They've been really great. To be fair, they've done a huge amount of work as well to prime the pump. So all the regulatory components are sort of in place now with this drug. And so the last piece of the puzzle is, unfortunately, you don't want to say things come down to money all the time, but
yeah, they do.
In this case, this is true now, that we're in a position where if we could get the capital, the funding to actually do these trials, we could get going almost immediately.
Really?
Yeah. We're hoping that obviously doing these types of media and talking to people like you raises awareness and, gives us that opportunity that someone out there will hear and, hear the call.
And if you want to help, QIMRB.edu.au, or to find out more about Professor Bryan Day and his group's work, QIMRB.edu.au. Thanks, Bryan. Congratulations.
Yeah, thank you. It's always a pleasure to talk to you, Claire, and really glad to share what is a really positive story, not just for us, but a whole bunch of scientists and clinicians around the world. And more importantly, I think it gives hope for patients and families.
I've seen you doing media over the past couple of days and you can't wipe the smile off your face. Well deserved.
Thank you so much.